when you say prokaryotes, these are cells without nucleus and without membrane-bound organelles. So these are cells that are very simple. Many of them are unicellular. That means they're only made up of one cell. There is no compartments. There's no different parts inside the cell, okay? Now, as for your eukaryotes, as animals, of course, we are eukaryotic. That means our cells have nucleus and our cells have the different organelles inside the body. Each of these organelles, of course, would play very important roles, okay? Now, as for your prokaryotes, again, your prokaryotes do not have nucleus. Their DNA, their genetic material usually is in a single loop, okay? That means it's circular DNA. Oftentimes, this is called a plasmid. Now, because of the difference in the structure and because of the difference in the way that their DNA is formed or the way that their DNA is arranged, your prokaryotes and your eukaryotes, there is also a difference in cell division, okay, in the way their cells will divide. Now, there is a very important part of the cell which we call chromosomes, okay? Chromosomes are rod-shaped structures made of DNA and proteins. Oftentimes we say chromosomes are condensed forms of DNA, okay? So these chromosomes, these structures contain your genetic information, okay? Whether you are going to look like your father, whether you are going to look like your mom, or maybe a combination of two of them. Maybe you are going to be as tall as your dad, maybe you'll be shorter, okay? Maybe you'll be short because your mom is short, for example. It all depends on the characteristics, the DNA, the information that you find in the chromosomes, okay? So again, chromosomes, these are condensed form of your DNA. That is DNA plus proteins, okay? Now your prokaryotes, since they do not have nucleus, again, we know that their DNA is just found in the cytoplasm, in that region which we call the nucleoid region. And because of this, they reproduce very simple. Okay, in a very simple process, which we call binary fission. This is what we find in your bacteria. Okay, they reproduce through binary fission. The DNA is copied and results into identical chromosomes. And of course, eventually, the cells would begin to divide. Bi there would mean two. Okay, so this means two, and fission, of course, we know that this means separation, splitting. Okay, so when you say binary fission, the form of asexual reproduction which happens in your prokaryotes, what happens is that the cells would just divide by two, divide by two, divide by two. And so eventually you are going to have a colony of your bacteria. That means you are going to have a group of bacteria. It's very fast for them to reproduce because the reproduction is just through binary fission. Okay, so cells would just divide by two, divide by two, divide by two. And so you are going to have a cluster of bacteria, which we call your colony. Okay, so again, in prokaryotes, cell division is just through binary fission. But of course, we know that in eukaryotes like us, it is going to be a more complicated process, okay? In your case, in our case as humans, of course, fetal development is very complicated. It goes through several processes. We all start with your zygote. Okay, when you say zygote, this is a fertilized egg. That is, of course, the union of the egg cell of your mom plus one of the sperm cells of your dad. Okay, now once the sperm cell of the father would fertilize the egg cell of the mom, what is produced, of course, is your fertilized egg, or we call this the zygote. Okay, now once your egg cell, the egg cell of the mom is fertilized, it is going to undergo several differentiation, okay, several divisions. It's not as simple as the binary fission that we've just seen in your bacteria. It is going to be divided into 2 cells, then 4 cells, and 8 cells, 16 cells, and so on, until it becomes a blastocyst. Okay, blastocyst here is very important. I will tell you why this is important in our discussion today. Your blastocyst is around 5 to 6 days of your fetal development. Okay, so just 5 to 6 days of a fetus. Okay, that means a zygote, it already has formed your blastocyst. Okay, when I was pregnant, I went to the doctor maybe, I cannot remember, maybe by 4 weeks of pregnancy. And I can see that my baby inside my womb, she already had a heartbeat. Okay, so just very early, at a very early stage of fetal development, that is already considered a baby, okay? It's already considered a human. It's a miracle, of course. It's a miracle of life. And we know that it has gone through different differentiations, okay? Several processes until, of course, you are formed by 5 weeks. This is what your fetus would look like. By 40 weeks, the fetus would look like this. Not a lot of pregnancies can reach up to 40 weeks. Some of them would just be by 38 weeks. I gave birth at just 38 weeks, okay? I did not reach 40 weeks, okay? My baby did not reach 40 weeks. I gave birth to her by just the 38th week, okay? So 20 weeks, this is what the fetus would look like. 10 weeks is just what the fetus would look like, okay? So until such time that, of course, you have the newborn baby, it's a complete human being, and of course, we know that it is a miracle of life, and that baby has gone through several stages of development, several stages of differentiation, and this is what we are going to be talking about today. Okay. Now, when we say cell differentiation, that very important process that made you into what you are today, this is the process by which cells become specialized and take on specific roles in an organism. So we all know that we all started as a zygote, that is a fertilized egg cell, and that egg cell is going to be divided into 2 cells, then 4 cells, then 8 cells, and so on, until such time that these cells have already gone differentiation. Okay, and when we say they are done with differentiation, they are now going to take on different functions. Okay, they will look differently from each other. There will be nerve cells, your neurons. This one would of course be found in your brains, and you are going to also become you will also have your epithelial cells. These are found in your skin cells. So from that single cell, a lot of different cells are going to be formed, and each of these types of cell would have different functions. Okay, now this is directed by the genetic information encoded inside the cell's DNA, that genetic information that is passed from parents to offspring. Okay, so again, that's cell differentiation, the process by which cells become specialized and take on specific roles in an organism. Now, there is a very special type of cell, which we call the stem cell. This is our topic today. This is very special because whenever you have a stem cell, you can actually direct it. Your scientists can actually direct your stem cell, can actually develop this into any types of cells that they'd want to develop this. So your stem cell can be placed in the lab and can be placed into different conditions until they become a muscle cell. They can become a fat cell, a bone cell, blood cell, and the rest of the cells that your scientists may want them to become. Okay, so again, stem cells are very special types of cells because they can be guided through their environment to the different conditions that your scientists can put them into, and of course they can be formed into different types of cells. Okay, so they can be directed to become differentiated into different types of cells. Now, your stem cell, again, this is very unique. This is a very special type of cell. And it can become 2 types, okay? It can perform 2 functions. It can have self-renewal. When you say self-renewal, it can copy itself, okay? It can make copies of them, of itself. So you can make identical stem cells from a single stem cell. Another process that it can do is, of course, differentiation. Okay, so your stem cell can become specialized cells. You can form this into a nerve cell. You can form this into a muscle cell, into your skin cell, any other cells that you'd want it to become. Okay, and so we say stem cell is very special because it cannot only become an identical stem cell, but it can also be differentiated into different types of cells. Okay, now, so again, we have self-renewal. We've already talked about this in the previous slide. So that means when you say self-renewal, your stem cell can just become another stem cell. It can maintain the stem cell pool, or it can also become differentiated. That means it can also become a specialized cell. It can differentiate. That means it can also replace dead or damaged cells throughout your life. So you may have heard the term stem cell when you hear news about celebrities, for example. If they want to keep their youthful faces, for example, their youth, they are going to be using stem cells. The stem cells get injected so that they still replenish those cells that are dead or those cells that are damaged throughout their body. The what? Yes, so your dead skin cells, for example, your if you have a celebrity, for example, is already very old, they can inject new stem cells that have been changed into skin cells, and then they can look young again. Okay, so that's your stem cells. Very special because it can be differentiated into different types of stem cells. And not only that, if you have someone, say, for example, that person has a damaged liver. Okay, your doctors can make, your scientists can make stem cells, into liver cells, and those liver cells can be injected into the body of that sick person. Okay, so stem cells again are very important. It's a breakthrough in the medical area. Okay, now these are some very important vocabulary that we need to learn today because we are going to the different types of stem cells. You have the term unipotent. Okay, when you say unipotent, this can only differentiate into one type of specialized cell. So if you have a stem cell that is unipotent, that means it can only become one special type of cell. If you say multipotent, okay, when you say multipotent here, it makes, or it can make multiple, that means more than one type of specialized cells. So if you have a stem cell that is multipotent, you can make it maybe into a skin cell or maybe also into a nerve cell, but there still is a limit, okay? It cannot become any type of cell that we have. Now, when you say pluripotent, this can make most of the specialized cells in the body. Okay, so you can virtually make it into any types of cells that you can think of. That's pluripotent. Now, totipotent here, this is pluripotent plus cells that are needed during development of the embryo, such as your placenta and your umbilical cord. Okay, so your totipotent, that is the highest form of your stem cell. That means it can become just not just any types of cells in your body, but it can also become a placenta. It can also become an umbilical cord. Okay, those parts of a pregnant woman's body that can, of course, support an embryo. Okay, so these are different vocabulary that we need to know. And we are going to encounter this as we talk about your stem cells today. Now, these are the different types of stem cell that we have. You have three different types of stem cell here. Embryonic stem cell. You have your adult stem cell, also called somatic stem cells. Somatic here means body cells. And of course, you also have your induced pluripotent stem cell. Okay, now their sources are also different. The source of your embryonic stem cell, of course, is your embryo. We've talked about the blastocyst today, and we said blastocyst, this is a 5 to 6 day old embryo. Okay, so 5 to 6 day old embryo, very young human being inside the body. Some of the stem cells of your fetus inside the woman's body, inside the mom's body, can be taken out, and that can actually become embryonic stem cell. Okay, now characteristic of your embryonic stem cell, since it is still embryonic, it is pluripotent. That means it can be differentiated into different types of cells, okay, into any type of cell that you can think of. Now, your adult or somatic stem cell, the source of this would be the tissue of your fetus and also even that of the adult. Okay, so tissues from fetus and adult. Now, characteristic for this is that it is multipotent. Okay, not all, but it can also become several types of cells. Okay, several types of differentiated into several types of cells. Now, your induced pluripotent stem cell, this is the one that your scientists would make in the laboratory. Okay, so this is a cell from an adult which is cultured in the lab, and this is actually totipotent. Okay, that means it can become into different types of cells that we have in our body. Not only that, it can also be formed into the placenta. It can also be formed into the umbilical cord, which are, of course, very important factors, very important parts in pregnancy. Okay, so some people would argue that there are ethical issues with regards to stem cell. Okay, we are not, some people would say, we're not gods. Okay, we are not God, and so we cannot do this. Okay, because of course, through your stem cell, you can perform cloning, you can make an organism, you can make a human person, okay, a human being out of a cell from an adult. Okay, so some people would say we shouldn't be doing this. This is against the will of God. There are ethical issues surrounding your stem cells. Okay, any questions? Any questions, people? Okay, if you have no questions, let me go to the next slide. I'm pretty sure this is the last slide that we have today. There are some factors that can affect cell differentiation. Of course, the first one would be DNA. Okay, so it depends on the DNA that your cell has. Your DNA can direct the type of cell that your cell can give rise to. It can also be based on environment. Okay, so these two factors would affect cell differentiation. DNA, genetic information, of course, the environment. Say you have only one type of plant here, but they can give rise to different flowers, different numbers of flowers, different flower types, depending on the environment that you subject them to. Okay, so environment also plays a very important role in cell differentiation. So again, don't forget, it's not only the DNA that can become a factor in cell differentiation, but also environment. And so our scientists can make your stem cell into different types of cells that they can think of because your environment affects cell differentiation. Again, when you say cell differentiation, this is a process through which a cell can take different roles, can form into different structures. And so, of course, we know that we are all what we are right now because of cell differentiation. We all started from a fetus, okay, from a zygote, a fertilized egg cell. That egg cell has developed, that egg cell has gone through several processes until we became fetuses. And of course, we came out of the bodies of our mom and we become what we are right now.